So let's now talk about the anatomy of the lens. So we just have mentioned that the cornea is forming about 2/3 of refractive power of the eye, and finally about 30 or 35% of the refractive power of the eye formed by the lens. So what is the lens? Lens is a transparent bioconvex structure that is located behind the iris and pupil and anterior to the lenticular fossa. So this is the lens. It's called the crystalline lens. So if you want to identify the lens, you say that it is a crystalline lens located just behind the iris and the pupil in the what is called in a depression within the vitreous cavity into the anterior vitreous called the lenticular fossa that we will mention about the next lecture in the vitreous. So the shape of this lens is a biconvex, as it is called the lens. So it is lenticular in shape, having a biconvex structure, which is different from the cornea, which has a convex anterior surface and a concave posterior surface. But the difference between the anterior surface and posterior surface that the anterior surface is less convex. So there is an inversional proportional ratio between the radius of curvature and the convexity. So for the more curved the surface, the less radius of curvature, while the less convex, the more radius of curvature. That's why the anterior surface has a less convex surface, but it has a radius curvature of 10 millimeter, while the posterior surface is more curved and has a less radius of curvature. It's about 6 millimeter. This relation between the curvature and the radius of curvature will be discussed more in detail in optics. So again, the anterior pole is called, it is less curved and has the center of the anterior pole of the anterior surface is called the anterior pole, while the posterior surface is more curved, as you can see, and the center of the posterior surface is called the posterior pole. Then the lens has an equator. So this equator, this is the equator of the lens. And the equator is separated from the free edge of the ciliary body by about 0.5 millimeters. So this is the ciliary body. Here it is. And the lens is separated from the ciliary body by about 0.5 millimeters. So what held the lens in its position? There are what is called suspensory ligaments. The suspensory ligaments are called the ciliary zonule, or zonule of Zen. They are attached the lens to the ciliary body. That's how they maintain the lens into its position, just behind the iris and in the lenticular fossa. These zonular fibers that arise from the ciliary body fuse to the equator of the lens, and they extend into the equator more into the anterior surface than in posterior surface. So if we consider this is the ciliary body and this is the ciliary process from which the zonule are attached, they adherent to the lens capsule in the equator region, more extending more anteriorly than posteriorly. Diameter of the lens is variable with age, meaning that the lens may be the only ocular structure that increase in size during all the years of life. So it doesn't stop in growth. So at birth, it's about 6.5 millimeters, while it reaches the adult size, become 10 millimeters. Thickness of the lens will be different according to the accommodation state, meaning that if the lens is accommodated, this will lead to anteroposterior bulge of the lens, leading to more curvature of the anterior and posterior surface, leading to more power of the lens. So the lens will be more thick. While in the unaccommodated state, the lens will be, the anterior and posterior surface will become more flattened, so the thickness of the lens will decrease. So here you can see in unaccommodated state, the lens is flattened by the traction by zonule, causing flattening of the lens, leading to decreasing in the thickness, while in case of accommodation, the zonule will relax, leading to the lens will bulge anterior and posterior by the effect of elastic capsule and the plastic lens substance. So this will be discussed in details in accommodation in physiology. So just I know that there are some new information too much for you today, so I will not go to further details for everything. So I will just stick to the anatomy. The refractive index for the lens is different according to if is it a cortex or nucleus. So the cortex is 1.3 and the nucleus is 1.4, and this will again have important aspects in the optics. Dioptric power of the lens is about 15 diopter, and the accommodation power of the lens will be increased more at the younger age. So at birth it is about 15 diopter to compensate for the small axial length. And as we get older, so the axial length increase and the dioptric power of the lens decrease. So it become 8 diopters at 25 years old, and at 50 years old it become only 1 to 2 diopters. So as we mentioned, that the lens continues to grow or the entire life. So the lens, the weight of the lens is about 65 gram, that increase up to with age maybe up to 220 grams. Color of the lens again is different according to the age, meaning that in infants and young adults it becomes transparent, while as we get older the lens acquire a yellow hue that become a yellowish tinge at the 35-year-old. With old age become amber yellow, very old become gray, what's called the nuclear sclerosis, due to impairment of antioxidant mechanisms that we will mention about again in the physiology. So we'll know why the lens gets opaque as we get older. Here we come to the microscopic picture of the lens, and here we come to maybe important anatomical facts that need to be studied thoroughly. So we have the lens capsule. The lens capsule is highly elastic. And what is the elastic? We will say this in the physiology. What's the difference between elastic and the plastic? This is not today. So the lens capsule is the capsule that will cover the lens total, and that doesn't contain true elastic fibers. That is made just by fine fibrils. So this is the important anatomical fact that you need to know, that although the lens is elastic or the lens capsule is elastic, but it does not contain true elastic fibers. With the electron microscopy, it has an lamellated appearance, so it appears like an onion. So it has layers over layers over layers, appears by electron microscopy, and by light microscopy, it is periodic SHF positive, more thick anteriorly than posteriorly, with the thinnest location on the posterior pool. This is very important and very frequently asked in the exam. So the thinnest location of the capsule is the posterior pool. So here is the lens capsule, which is the black one. And as you can see here, it is thin, or the most thin part at the posterior pool of the lens. Just beneath the lens capsule, there is a single layer of cuboidal epithelium. It's called the lens epithelium. So lens epithelium, it is a single layer of cuboidal cells just beneath the capsule. So is it cuboidal all over the epithelium? All over the entire surface of the lens? The answer is no, because the lens epithelium is cuboidal at the center, just at the center, and they rarely show mitosis. So the lens in the epithelium of the center is not metabolically active. However, as we go toward the periphery in the intermediate zone, the lens becomes, or the cells of the epithelium becomes more narrow and more elongated with increasing mitosis, and so the lens epithelium becomes more active. And here we come to the equator, where the lens show the maximum elongation and show the maximum mitosis, and this is because this equatorial lens epithelium will form the lens fibers. So basically the epithelium is just beneath the capsule, and the equatorial epithelium will divide later on to form the lens fibers that will be compressed further and further and further to form the nucleus and the cortex. The capsule is the basement membrane of the capsular epithelium, and there is a very important fact that we'll know in the embryology, that there is no posterior epithelium. So as you can see here, the posterior epithelium is present, and it present at the area of equator. But in case of the posterior, there is no posterior epithelium. And why is this? This is an embryological fact because the posterior epithelium was consumed during formation of primary lens fibers. Again, the posterior epithelium no longer exists in the adult life because the posterior epithelium is consumed during formation of the primary lens fibers. So after we know about the lens capsule and the lens capsule and the lens epithelium, here we come to the lens cells or lenticular cells. So as you can see here, this is the lenticular cells or lens fibers are acquiring a laminated appearance. So the deeper cells, the cells or the fibers are formed at the equator, pushing the older fibers toward the center so that the newer fibers are more superficial while the deep fibers or the old fibers are more in the center. So in the center, it is formed by the nucleus, and the nucleus is formed by the old lens fibers, while the superficially it is the cortex and it is formed by the recent lens fibers. So we have a comparison between the immature lens fibers which are located superficially and the mature lens fibers which are located more deep. This is very important and has many important data that is asked in the exam. So let's have an idea about them. The immature lens fibers which are located near the equator are smooth because they are well hydrated, because they are near to the aqueous. So the lens fibers, the immature lens fibers present near the equator are smooth and well hydrated. On the other hand, the mature and lens fibers which are more compressed toward the center are away from the aqueous, so they lose their fluid and they are becoming less hydrated. That's why they become serrated edge. Their edges become serrated due to decreased hydration. The immature lens fibers consist of, so this is a lens fiber. It consists of a nucleus, anterior process, and posterior process. So this anterior and posterior process or the posterior process are in junction with or attached to the posterior lens capsule, as you can see here. But as the lens fibers are compressed toward the inside, as you can see here, the mature lens fibers become losing their attachment to the posterior capsule. So here is the superficial fibers or the immature lens fibers, and here are the mature lens fibers. As you can see here, the mature lens fibers has some changes. One of the changes is that it lost its attachment to the posterior capsule. The other thing or the other change is that the mature lens fibers lost their nuclei. So you can see that the immature lens fibers have nuclei, while the mature lens fibers don't have nuclei. So they lost their nuclei and they lost their attachment to the posterior capsule. Other thing that is characteristic for the immature lens fiber, it was called the nuclear bow. So the nuclear bow, it is like a curvature or a curve of made by the nuclei of the immature lens fibers near the equator as the compressed or as the oldest fibers are compressed inside. So as the fibers are compressed, the nuclei migrate, meaning that they will forming a nuclear bow. So it is frequently asked in the exam, what is the nuclear bow or which type of cells forming the nuclear bow? So it's formed by the immature lens fibers. So for the mature lens fibers, the anterior process and the posterior process are joined by a specific cell junction. These cell junction are called ball and socket and tongue and groove. So as we said in the cornea, we have specific cell junction like desmosomes, zonula occludens, and hemidesmosomes, but the lens fibers have specific types of cell junctions called ball and socket and tongue and groove. As you can see here, this is the junction between the lens fibers together. This can be in the form of tongue and groove, as you can see here, or ball and socket. So ball and socket and tongue and groove are the specific cell junctions for the lens. So from this we know that the cortex is formed by the superficial lens fibers from the immature lens fibers that have their nuclei near the equator forming the nuclear bow, and their posterior process are attached to the posterior capsule, while the nucleus is formed from more mature lens fibers that lost their nuclei and lost their attachment to the posterior capsule, and they are more rigid and more with serrated edges and more compressed centrally. The junction between the lens fibers together will make a suture. So anterior suture will be in the form of an erect Y suture due to apex positioning of the lens fibers, while posteriorly the fibers are joined in a manner of again Y-shaped suture, but it is like of the Mercedes sign, so it will be an inverted Y suture. So due to base opposition of the lens fibers. So this is asked in the exam that the anterior Y is due to apex. So remember A for A. Anterior Y or the erect Y in the anterior surface caused by apex opposition of lens fibers, while posterior Y, which is inverted, caused by base opposition. Here is the Y suture. This is the anterior Y suture. It is an erect Y, while the posterior Y is inverted. So this is the electron microscopy section of the lens, will showing this is the anterior lens surface, showing that this is the erect Y anteriorly due to apex opposition here, while this is the posterior lens surface, show the Y inverted Y-shaped suture due to base opposition. And the important clinical aspect of this Y suture, that sometimes it can be opacified, leading to what is called sutural cataract. Here is the sutural cataract. As we mentioned, the lens continues to grow over the life. So the first nucleus the first lens to be formed in the embryonic life is called the embryonal nucleus. The embryonal nucleus is formed in the first 2 months of gestation, and later on it is covered by the fetal nucleus, and the fetal nucleus will be formed from the second month till the last month of gestation. Then other lens or it will be new fibers lay down over the fetal nucleus, will form the infantile nucleus, which continues to grow till the 25 years old, followed by finally the adult nucleus, which is formed after 25 years old. Blood supply of the lens. So the lens is a vascular structure, and this is important to maintain transparency, but the lens will be supplied during the embryonic life by an artery that is formed during the embryonic life and disappear later on, which is called the hyaloid artery. So the hyaloid artery will form what's called the tunica vasculosa lentis, which is a vascular bag that would enclose the lens and support the growth of the developing eye, and this will be discussed in the embryology. Changes with age that occurred with the lens will be discussed in the physiology. So the lens will be more growing. The lens continues to grow throughout life, becomes more flat due to decreasing accommodation, with decreased rate of new fiber formation, increasing the thickness of lens capsule with a change in the color to yellow, and development of senile cataract. So is everything clear up till now in the lens? Okay, so let's talk about the suspensory ligament of the lens, or what are the structure, or what is the structure that hold the lens in its position. This is called the suspensory ligament of the lens, or ciliary zonule, or the zonule of Zinn. So the ciliary zonule or the zonule of Zinn consists of fibers that pass from the ciliary body, or specifically the ciliary process, and attached to the equator of the lens, leading to holding the lens into its position. The distribution of this lens fibers or this zonular fibers will make two important canals or two important anatomical spaces within the posterior chamber. The first one is called the canal of Hanover. So the canal of Hanover, it is the space between the anterior membrane or posterior membrane. So the zonular fibers makes an anterior membrane and the posterior membrane. The space between both of them, this is called the canal of Hanover. While canal of Petit, this is called canal of Petit, which is the space between the internal membrane or the posterior membrane and the anterior hyaloid face. So you can just remember it like anterior and posterior. The anterior part or anterior space of the zonule is called the canal of Hanover, while the posterior part or the posterior space of the zonule is called the canal of Petit. According to the electron microscopy, we have three types of the zonular fibers. So we have type 1, type 2, and type 3. Type 1 is 1 micron in diameter and usually near the vitreous, while type 2 is more thin and flat. Type 3 is the most thin in all types of the zonular fibers and have a circular course. So we need to know some data about the types of zonular fibers. So we have main zonular fibers and the axillary fibers. For the main zonular fibers, we have first the orbiculoposterior capsular fibers. What is the orbiculoposterior capsular fibers? These are the fibers that forms the most and the inner part of the zonular fibers, attaching the ora serrata to the vitreous. So here are the what's called orbiculoposterior capsular fibers. These are types of the zonular fibers that attach from the ora serrata, which is the most anterior layer of the retina, and they come in close contact with the anterior limiting membrane of the vitreous, and then insert into the vitreous or what's called the ligamentum hyalidocapsular of Wigert. So this is the capsulohyaloid ligament of the vitreous, and this will be discussed in the vitreous, so you will know what is this hyalidocapsular ligament. So again, orbiculoposterior fibers are type of the zonular fibers that originate from the ora serrata, which is the most anterior layer of the retina, and pass in close to anterior limiting membrane or the anterior limiting layer of the vitreous. So it will pass over the anterior surface of the vitreous till it insert into the hyalidocapsular ligament with the thickness of the thin type, which is the second type. This is called orbiculoposterior fibers. So as we said, there is orbiculoposterior, there will be orbiculoanterior fibers. So orbiculoanterior fibers are arise from the orbicular ciliaris or the ciliary body, and they occur or they locate in the valleys in between the ciliary process, and they are inserted into the anterior surface of the lens. That's why they are called anterior capsular fibers, and they are the thickest and the strongest. And here what is asked in the MSQ. So the orbiculoanterior, as you can see here, it is the more thick and the most strong. The cilioposterior capsular fibers arise from the valleys in between the ciliary process and pass and insert directly into the posterior capsule. And they are the most numerous of the zonular fibers. So they arise from the ciliary process, from the valleys in between the ciliary process, and insert into the posterior capsule, and they are the most numerous. So the cilioposterior is the most numerous, and while the orbiculoanterior are the most thick, where the thickness is belonging to the most thin part. Cilioequatorial fibers, so they arise again from the ciliary valleys, and from its name, they are inserted into the equator. So they are present in young age, and with age, these fibers disappear, and few only remains, and the thickness is of the third type. and insert into the capsulohyoidocostal ligament. So they pass over the anterior surface of the vitreous to be inserted into the most anterior condensation of the vitreous behind the lens, which is called capsulohyoidocostal ligament. Then we have the orbiculo anterior, which is the most thick and arise from the orbiculus ciliaris and insert into the anterior surface of the lens. We have the cilio-posterior fibers, which arise from the ciliary body and insert into the posterior surface, which are the most numerous and thin. And we have finally the cilio-equatorial, which arise from the ciliary body and insert into the equator. There are some fibers that will support the main fibers are called the auxiliary fibers. So they just support the main fibers of the zonule together, and in order to prevent their separation of the two neighboring process. So the two main parts of the auxiliary fibers are the orbiculociliary fiber, which are run from the orbiculus ciliaris to the posterior ciliary process, and the interciliary fibers, which join the ciliary fibers together, preventing their separation.